Thank you all for joining us for our webinar, which will be presented live by our guest speakers, Jean-Marie Galano and Ángel Sánchez Illana, entitled Unveiling Oxylipin Diversity, a journey from MSMS library implementation to molecular networks. My name is Maria Fedorova, and I'm based at the Technical University of Dresden in Germany. This webinar is being recorded and will be uploaded onto LIPIDMAPS website and our YouTube channel within the next 48 hours. The webinar will be around 45 minutes, allowing 10 minutes at the end for the questions. Please use Q&A function through the presentation to ask your question, and we will unmute you to ask your question directly to speakers at the end of the presentation. Today's webinar is being sponsored by Avanti, and we are grateful for their ongoing support. Let me introduce now our speakers for today. So Jean-Marie Galano is the Director of Research at the National Center of Scientific Research, CNRS in France. He studied chemistry in the University of Marseille and obtained his PhD in 2001 in the field of natural product synthesis. Then he moved to the University of Oxford to pursue his postdoctoral fellowship for 3 years under Marie Curie Fellowship, and then joined CNRS in 2005 as a researcher at the Laboratory of Synthesis of Bioactive Lipids in Montpellier. His research initially focused on the novel chemical approaches for the synthesis of oxylipins, especially isoprostanoids and isofurans, and currently he also contributes with his expertise in organic chemistry to lipid and lipopeptide research project together with Nicolas Sénac and Justine Bertrand-Michel in Toulouse. Along the years, Jean-Marie developed a very strong interest in application of mass spectrometry for the detection of lipids and oxylipins, and recently in the development of molecular networking to the field of non-enzymatic oxylipin analysis. He also loves to play with the quantum chemistry to understand reaction pathways or mass spectrometry fragmentation mechanisms, the tools he learned during COVID lockdown. And he also a fine adept of salsa. So if you meet him at a party or the conference one day, do not hesitate to ask him over for a dance. Our second speaker is Ángel Sánchez Illana, who studied chemistry and got his master in experimental technologies at the University of Valencia in Spain. His PhD's chemistry was completed at the Health Research Institute La Fe in 2021. Through his academic journey, he developed a robust expertise in GC-MS, LC-MS, and vibrational spectroscopy like FTIR and Raman, as well as chemometrics, with a specific interest on application and clinical and biomedical settings. Since 2022, he been a postdoctoral fellow in Montpellier, working together with Jean-Marie to enhance non-targeted analysis of oxylipin and novel non-enzymatic oxidized lipids derivatives using molecular networking techniques. So the stage is yours, Jean-Marie and Ángel. Please share your slides.
Okay, thank you very much, Maria, for the introduction of both of us. And we are very happy to show you what we have done for the past 2 to 3 years. Actually, this is the part of the PhD work of Anis Eloumi. So here is Anis Eloumi, and these are the papers that we submit in Cambridge, and now it has been accepted in Scientific Data. So pretty soon you should see it, and we go into more details. So here's the lab in Montpellier. If you guys want to come some days, so we have a very nice building, only one big. So in the lab, what we do oxylipin synthesis. So we are hardcore of chemists. We do quantification of those oxylipins, and we do as well many things with lipids conjugated, lipophenol conjugate, lipopeptide conjugate. So we have a big team. Well, we have medium teams, but we are interested in a lot of things related to lipids. And as you all know, we have very strong interest in oxylipins. And the definition of oxylipins was made by my big hero, Mart Hamburg, and he called this as being basically metabolites enzymatically formed from PUFA. So you have to employ a mono or dioxygenase catalyzed oxygenation to make these oxylipins. After they are released, after the PUFA is released from the phosphomembrane, by phospholipase. Then release of the PUFA, then different enzymes, you all know those examples, the COX, the LOX, the the CYP 450 can make a myriad of metabolites. I won't go into all those compounds. This is not really the purpose, the biological activity of those compounds, not even the quantification is really the matter here in this explanation. So I will skip the interest of these compounds, but you all know this. You've seen the talk by Ginger and Niels, the last ones. They are very great talks, and all the information you could retrieve there. But what else do we have? We've seen 3 particular enzymes, but there is as well the oxidative stress phenomenon that happen in lipid membranes. This oxidative stress will go into the generation of a high quantity of reactive oxygen species, to simplify this, and this will start to produce what we called in the lab, well, actually Marie Demion called this as a neo-PUFA. So they are non-enzymatic oxygenated metabolites of PUFA. And those are formed by the ROS inside the membrane, so still attached to the phospholipid. Your arachidonic acids or whatever will transform into something similar to an oxylipins. Later, it will be released by phospholipase to form end products, the neo-PUFA. And there is a myriad of compounds, much more than the enzymatic version, so the oxylipins, because of more isomers, obviously, and as well more chemical processes, probably. Here in the lab, we do quantification, like I say, particularly because they are like phospholipids, neo-PUFA, so you have to release it from the membrane, like Niels showed. This is an important process. You have to be careful with it. We do Q-trap quantification. Actually, we use it as an MRM, as a triple quad only, but this is what we do in the lab. We do quantification of oxylipin related to oxidative stress. One particular example I always like to show, well, for the last 3 years, is that example from the group of Craig Willock in the acronylic scarcity, where you clearly show that the isopostanoid or the neo-PUFAs are not just artifacts, like Moreau used to say. They are not artifacts of, PUFA oxidation or so on. They're really produced in a high quantity. At least here you can see as much as the PGD, for example, or the F. So this oxidative stress is related to a lot of diseases, right? So this we know. It has the oxylipin has deleterious effects. Okay, we know this. They can be used as biomarker of oxidative stress. This is what we do in the lab, and a lot of people do it. And those are really used as the clinical settings, like Ginger told us last time. They do as well bioactivity, biological active compounds. They are, having good effect, obviously, to the contrary of deleterious ones. In the lab, with a great team of cardiophysiologists led by Jean-Yves Le Guenec at the time, now Marie Demion joined the team, they show that the more you oxidize DHA, the better it seems to be for your cells to not be in arrhythmic events, right? To summarize this. We found by just luck that we made this compound before even think of this. And that compound was the neuropostin, the one I described here on the right part. That was just basically luck. Later on, we showed that this compound has an effect on the ryanodine receptor, and now we have a synthetic version that will go probably into a clinical phase of development in the next year to come. And this is another pathology, not antiarrhythmic, but the diaphragm mechanical ventilation injury. So this is something else that I wanted to show about. But I want you to show or to explain is, let's say now they come to us again and they say, oh, we found another DHA oxidation that is working on another ion channel. But it's not your standard. How will you do to find it? Well, this is a bit annoying because probably one of the only way to do is to do bioassay directed fractionation. But at the first step of this process, you will be stuck with that fractionation, that extraction, and that separation of the myriad of isomers. Here, I represent what's oxylipin in terms of formula. There's 168 formula, but only 25 of those formula represent a large majority of the molecules, and especially the non-enzymatic ones. So clearly, this is not something possible. Almost at the same time, there was a talk in France by Erwan Poupon that showed that you could do the replication study. You could use mass spectrometry, MS2 data to link metabolites all together to retrieve information that was known in the past. This is what they call molecular networking. This was done by the group in San Diego by Peter Dorenstein at that moment. So that was very exciting. And then at the same moment, they even show that you could link this, MS2 information and network with a bioactivity assay, right? And so in theory, you may be able to retrieve using statistical analysis on top of it, the bioactive compound. I say, perfect, let's do it. Let's dereplicate oxylipin. Almost at the same time, a paper came out, which was the perfect compound paper for us to get the grant that we wanted, because they show that you could dereplicate oxylipins, that you could basically retrieve information that was known because you have the standard. So they took a plasma samples, and what they basically did, they tried to retrieve information. They tried to retrieve similarities of this standard that you can buy. And they found like 500 unknown compounds, or putative compounds like they call, which was great because it was basically what we wanted to do. What I like to say as well is that if you look at the MS1 of an oxylipin at plus or minus 3 ppm, there's almost nothing else in the human metabolome as an oxylipin. So the MS1 is already a quite nice information to have if you're looking for oxylipin. Okay, one more thing. For dereplication, for molecular networking, what you need is a standard. Okay, good. The neopufa are very similar to oxylipin. Some of them, so they have the same, similarity, which is great. This is what you need. But there is a lot of known neopufa, non-enzymatically formed, that we don't have any MS2 data. And Simon Hamann showed this in that Twitter link where they say it's quite messy, the field. Well, actually, yes, indeed. For example, if you take that particular compound, this is a dioxoline unit, which is formed during the process of the peroxidation. So you have this radical that can form a myriad of intermediate, goes all to the the isothromboxane structure that was discovered by Moreau. So in theory, this compound is known. You should have MS2. It's easy for the 15 series because it's the same as the thromboxyl that we all know. But what about all the other ones? We don't have any data except those ones that we made in the lab. All those compounds are being detected, but we don't have any MS2 data. And this is a particular importance because Valerie O'Donnell identified putatively a compound that they isolated in the past, being that dioxoline compound on the left, right? So basically, the intermediate are just shown just a bit before. And they showed that 3 years later that actually there was not that compound. So they did all the synthesis of both compounds, and they showed that there was actually this bis epoxide compound. The fun fact, or maybe not very fun, is that this compound was actually, found 10 years before, but the MS2 data was not available. So what I wanted to say, if you had data before, you could have retrieved it faster. Therefore, the importance. So therefore, what we need to do at that moment, we needed to construct neopufas. We need to oxidize as many polyunsaturated fatty acids as possible to have a lot of data, to analyze this data, and to do the replication and molecular networking. So for this, we use chemical tricks. We use oxidation, and we call it Porter tricks because Porter did it all. Yeah. So we basically oxidize PUFAs, ester of free acids, with H2O2. This was to mimic the biological assay of the cardiophysiologist, and we use V70 radical initiator with or without NMBHA, which is this hydrogen atom donor that Porter described, to block the peroxidation process somehow, to stop the endoperoxide formation or the cyclic formation. Then you will collect the data, and you purify it, and then you can do further purification or further transformation. So obviously you will collect the first one, you can analyze this, and you can further treat with triphenyl phosphate, where you will reduce the hydroperoxide into alcohols. But the funny fact that you will not reduce the cyclic endoperoxide, that one, but you will reduce this one. So using that chemical tricks, you may be able in your future network to know more or less what you will gain in the end, right? And we as well try this potassium hydroxide protocol that we have in the lab for saponification to check whether or not this non-transformation from the endoperoxide will transform into the ketone. Actually, our protocol does not, which is something that was good to know. So I should have put a bar. So now Ángel is going to take the mic.
Yeah. So, yeah, that is our general workflow that we use for the analytical part. So, yeah, in this schematic, you can show that we measure the analytical standards and the oxidation PUFA that Jean-Marie was talking about to make two things. On the one hand, a big library of MSMS data, and then the MSMS data to make the molecular networks to be able to carry out all the replication studies. All of this data, the library that we made with the standards and the spectra extracted from the oxidations are public in the repositories that we describe in the paper. So starting with the library, we include 59 neo-PUFAs made in our laboratory. Isoprostan, neuroprostan, neurofuran, phytoprostan, phytofuran, diomoisoprostan, diomoisofuran. And they here in Montpellier, they made very well during all of these years. And 62 commercials from Cayman, including enzymatic oxylipins that are commercial, but also another isoprostan non-enzymatic that are also commercial. And we include the 2060 MSMS data that Walters also made public in their study that Jean-Marie was talking about before. So for the oxidation that we made with the hydrogen peroxide, this is the protocol that we designed. We start with a small scale, doing 5 milligrams oxidation of PUFA, doing 30 minutes at 40 degrees, and then we apply the different conditions with the hydrogen, with the potassium hydroxide, for example, to mimic the effect of the hydrolysis, supposing that compounds will be in the mixture after an hydrolysis step. And then we extracted as the normal extraction for the oxylipins, employing SP solid phase extraction and the common protocols for the oxylipin extraction. And we made the 3-methylphosphate that Jean-Marie described before. So we have, for example, in that case, for each PUFA, we work with DHA, AA, ELA, and EPA per PUFA. And we dilute these samples in mobile phase to inject in the LC analysis. For the B70, we made in a bigger scale, employing 50 milligrams of PUFA ester in that case. And we incubated during 24 hours, during 1 day, 36 degrees in a sun bath. And then we purify them with flash LC to be able to separate the unreacted PUFA. So this is in that TLC, you can see the spots of the non-oxidized PUFA and all of the oxidation fraction that includes all of the neo-PUFAs and oxylipins. In that case, would be neo-PUFAs because are not enzymatic. And also, we want to know that in the case of the NMBHA, we have also to separate the NMBHA prior to injection because that NMBHA is quite annoying for the cells. And if you inject it, you are going to maybe destroy the capillary of the cells or whatever because it's quite sticky and you have to take it out. And then this fraction we collected together and make the dilution as we made with the hydrogen peroxide part. And we have per PUFA in that case 4. With reduction, without reduction, and with NMBHA and without NMBHA. And also as a proof of concept application, we extracted oxylipin from 2 kinds of samples. One blood serum samples and another from algae samples. Follow the extraction procedures that we published in the past, the group of here in Montpellier. So, and the point was to see if we can use it, the networking approaches also with real samples in that case. What LC analysis we made? So we try, on the one hand, to make a FIA method to analyze the standards to obtain the MSMS data of the standard straightforward, just with the NAPIA injection. And for the analysis of the oxidation mixture, we make also a straightforward gradient using water and acetonitrile. And our idea was to make the method as simple as possible, because we know that all of these high-resolution machines that we use and thermo IDX, 3D mass spectrometer, are normally in common platforms. So it's more easy if the methods are straightforward, I think, in order to tailor, to adapt easily the method. So we use the collision energy steplet from 20 to 60 to collect as more information as possible in the MSMS data. And we use an inclusion list, so for picking the MSMS data. That inclusion list was made using the Jean-Marie knowledge, I want to say, because at the end, it's the oxylipin molecular formulas could be just adding oxygens and hydrogens and deleting hydrogens and oxygens, and using that rules that he knows very well, we can make just an Excel sheet or making an easy script to have all of the exact masses and then create an inclusion list of the oxylipins, and we can repeat that process for DHA, ELA, EPA, or whatever PUFA you want to use. So all of the samples that we analyzed with that method, we made it in a, for example, in the oxylipin, in the oxidized PUFA, in a sequence like a metabolomic experiment, using QCs, blanks, and so on, in order to provide a data set that the people can use for example, if they want to try some new algorithm for picking, because the people that work with, and also Jean-Marie told before, is one of the main challenges, the chromatographic thing, because it's very difficult to resolve sometimes that compounds chromatographically, and also to pick in the feature. So it's important to have this data set well defined. So we made it in that way. And also we analyzed in another day the serum and algae samples. So how the data analysis part, the library, we just collect the spectra and submit to GMPS. GMPS have a very nice spectra batch addition tool where we can just submit all the library, and we will have that kind of structure at the end where we can include the structures, and we can see also the MSMS and some metadata, and we can use all of this library in the GMPS workflow. They have a lot of workflow for doing different kinds of molecular networks. So the molecular network, the people that knows, it's based on the modified cosine similarity. They find a way to make a similarity between MSMS spectra and then to build a network that connects that similarity. And that modified cosine, it takes into account the pairwise comparison based on also the precursor ion differences. So if the precursor ion is different, you can align the spectra and select different, select the peaks with the precursor ion difference to the cosine score calculation. So in that way, you can take into account the neutral loss information in the network. So if we make that network with all of the standard that we have that kind of connections, and we can find some spectral similarities that we will discuss in some slides. So in the case of the oxidation mixture, if we want to extract all of the MSMS of that samples, we made the feature-based molecular network, because at the end we have features like this. For example, imagine this is one of the samples that we have, the extracted ion chromatogram of one of the features, the monohydroxide of the DHA, for example, and this is the MS1 and this is the MS2 spectra that we collect. So you can see that we have the elution profile in MS1 and some of the MS2 spectra. So we have to select the MS2 for each feature. So this is easily implemented with MSETMine very well. you can automatically extract the eggs, all the eggs of your sample, of your data. You can then deconvolute all the features, and then you can assign to each feature one MS2, or 2 or 3 or whatever. In that case, we have, you can see we have 2, 3 MS2 per peak, so we can select the most intense. And then all of this data across sample, we can align them. So we can align all the features that we have. That feature, I mean, the chromatographic peaks, phase, retention time, MZ in the eggs, and then we can align all of these features across samples of different PUFAs or across all the PUFAs. And at the end, we will have the table of features with a representative MSMS per each feature. So with this table, we can build a network, molecular network with the MSMS, but in that case, each node will be an MSMS and with quantitative information or semi-quantitative or intensity information. So we can build that kind of P charts when we can analyze the different proportion of signal under the different oxidation conditions. That is our main goal, doing all of these oxidations depending on the B70 and MBHA and so on. So the plasma and algae samples that we try to analyze in that way, we can do it exactly the same. We can extract the feature, and then we can build a different type of network. But we try, because the alignment could be more difficult, because at the end are different type of samples, measure different days, and could be difficult to align them. So we just extract all the MSMS data, and then we build a network with the MSMS data of the OXPUFA, the oxidized PUFA, and the algae and the serum samples. And then we can also assume some of that MSMS data doing the networks in that way. So yeah. Jean-Marie is going to go in deep in the networking connections.
I will try, because it's not an easy. So I will recommend you to read the paper, obviously, the ChemRix, but basically what we can, this is the Neo MSMS. So those are the standards that we made in the lab and the standard that worked by and provided to the community. So we made that library, and we can see connection. If you look here in the node, you can see that the furan families, okay, there's not a lot, but they're all together. But those are not very interesting. The one which are interesting is, for example, if you look at the linear compounds, they are more or less all clustered together somehow. The F-isopostanoids, okay, they are as well all together, and so on and so on. This is one representation that you can already see by yourself. Another one is like, if you combine the number of oxygen, except the carboxylic acid, you can see that they are as well somehow linked together. We think this is because of the dehydration process of the MS2 in your MS2 pathway. But you can as well see similarities based on the position of the allylic alcohol, that famous allylic alcohol, where the main fragments came out of it, right? The main MS2 data usually. So for example, here, number 7 are those compounds. So you can see that this is because they are all the same end part of the molecule. So, but that's quite easy, because this is only a standard that we made. So now this is OX-DHA. So all the oxidized DHA that we performed, that Ángel talked about, and all the different conditions. And here's the P chart that you can see. You can see some oxidation, you have one particular only compound, and some you have differences. So this is interesting, because this is actually what we know the best. This is the neuropostanes. Yeah, we have the standards. So you can see that each of these square compounds are some compound that we had. So they were in the network because they were in Neo MSMS library. So you can see they are here line, square, and you can see there is a lot of them. We have only one. All of them are isomer because they have chromatographic feature. They have a proper retention times. So you can de-replicate basically isomer of the 20 series of neuropostanes. What you can see as well, there is some that we don't know about. What are those compounds? So to understand this compound, you have to understand fermentation process of, for example, the F-isopostanoids. This was well known when described by Murphy. one interesting feature is that isoprostanoid, that particular deprotomer, is not losing carboxylic acid as everybody thinks or remember badly. It's like it's going to lose acetaldehyde. So this is it's going to be the same minus 44, but not quite so accurate masses, obviously. And then you lose water, CO2, etc., etc. Another deprotomer can lose water, CO2, and C2H4 most of the time, and as well those two. So these are basically the pattern that you will retrieve in each of the standard that we have, those green thing, right? This is the pattern. This is how the compounds are linked together there, actually. But what about this unknown one there? So it's easy. If you look at the spectra, you find the pattern again. But then you look at the main fragmentation. So there's only 3 main compounds that you know about, which are produced, the 7, the 17, and the 11. It's quite easy now. You just have to look at the pattern of the fragmentation. The stuff that you know best is how the compound is fragmented here at the allelic position. And you can retrieve the compound. And actually, the compound here is the 7 isomer. We don't know if the trans, cis, or whatever. I guess we don't really care at the moment, yeah? You being able to dereplicate. Same thing for a more complex set. This is the all oxidized DHA, EPA, ALA, and arachidonic acids. Now, how the compounds are connected, they are a bit different, I'll just show. Most of the time, they are now linked with the series patterns, right? So, for example, maybe there is not the best example so far, but you can see some of the 5 are linked to some of the 5 there, and as well linked to the 8 because they have quite a similarity in fragmentation. So everything now is, if you have a full real knowledge of what you have inside all the PUFAs, so quite similar to what you get in real sample. Now you know that the compound will be more connected by series and not really much so by families. This is more or less true because, for example, here, compound that we don't have the E4 neuroprosteins. Nobody really has it. Or if they made, nobody knows, and they never publish it or whatever, the MS2 especially. But those compounds are connected with the E series of phytoprostan. They are long shot, but still they are connected. And as well with the prostaglandin, the one which are commercial. Similarly, the E are connected to something which are the A isoprostans. Nobody really much published the MS2 data. There's no compound available to inject into your library. But because they have this fragmentation similarity, actually the spectra are almost the same, unless the precursor ion is not the same, right? Because they are, first thing is going to lose water. So this is how you can connect and understand your network. But this, I would say, is very simple because you have data. Remember, to do molecular networking and to understand your cluster, a cluster is this, a cluster is that, you need a standard. If you don't have standard, I would say you are on your own with fragmentation knowledge. If you don't know any of the Murphy rules, you're going to be in big trouble. Hopefully, you have the MS1 accuracy. You know about MS1, it's an oxylipin. You have that chemical network tricks that I talk about, the phosphate reduction and so on. Annotation software, they don't work. But we believe that the machine learning is going to be the next thing for our PUFA. We can discuss this in the end, and we are doing it. Not quite, but we know how to do it. But what you can do is you can make your own MS2 compound, right? Let's say you're a big fan, like us, of the leukotriene of Ned Porter. You know that Ned Porter oxidized the 15 hits with that V70 oxidation. He did this in a big scale. We did this in the microgram scale. We bought the standard from Cayman and we oxidize it, except we didn't put that hydrogen atom donor because, like Ángel said, it's going to mess our MSMS. So by doing this, so Porter found 7 compounds, no MS2 data. Well, actually, there is, I think, 4 MS2 data, 1 fragment for this compound, that one, and another one. We only could retrieve 4 compounds. Those 2 are commercially available now. And this one, because it gives the fragmentation, 1 MS2, we could retrieve and find the MS2 explanation. And then we were stuck into those ones. But then once again, what you have to do, you have to take your spectra and try to understand. So one of the reviewers say it's quite bizarre that you try to explain that fragmentation process by cutting a double bond, which is something that you don't usually do. This is not quite true because Resolve D1 is like so. Serran showed it in the past. But, if you know Murphy rules, everything can move, like the sigma proton shift is possible for oxylipin in the time your molecule is flying through the MSMS. It's millisecond. It has time to do sigmatropic shift. So in the end, you can move a bit the double bonds. If you move the double bonds, therefore you are able to explain those fragments. You can explain it in different way. I can write crazy stuff in mass spectrometry, but everything is on the Murphy rules, right? You can even use software. I say in silico software are not very good, but this is this is something else. Actually, QCXMS is a bit more than the stuff that we have, but it's still a complex way to use it. But here, for example, I did this fast for the show. I use one particular deprotomer, and I can find 3 of those. And I have the fragmentation process, which was not at all what I wrote here, right? But anyhow, we are pretty sure this is the compound. If you're interested in this compound, likely you might be able to synthesize it. Same way, the levuglandine. This is a very truly beautiful compound that can rearrange, do a lot of stuff in vivo. We were interested into the MS2 data. There is no data, but it is known by Salomon that you can take this PGH2, right? You can buy it 50 micrograms from Cayman, it's quite cheap, and you can put it in DMSO after 1 hour, 40 degrees. You should get 5 compounds: the PGF, the PGE, the PGD, the keto something, and in theory the levuglandine. No MS2 data available. Now we have 1 compound. And 1 compound was a bit bizarre because this compound has 1, 2 loss of water and a CO2 consecutive loss. This is in theory not possible to explain because you have 1, 2 loss of water and you have not quite a CO2 to lose. But if you know your Salomon rules as well, you know that levuglandine will rearrange somehow into lactol. And now you have another oxygen here. So you can lose 1, 2, and a CO2. And you can even explain this fragmentation. And everything else fits the book of Murphy and so on. So you can retrieve information if you need. Another thing that you want to do, you can retrieve information, you can reanalyze information. That famous Wartruth paper that I was talking about, the 2019 paper, where they found 500 new oxylipins. I mean, the people in the oxylipin fields, they should be crazy about it. New oxylipins, let's go. Well, they found like 6 or 7 compounds which were actually correlated with inflammation, but they did not quite annotate them at all. Now it's fairly easy to do with OXPUFA, the stuff we provided. For example, this compound here they found, it matched very well with the 14-HDHA. If you work a little bit around, you have that very even masses, which is quite characteristic of this oxycop rearrangement. And in the end, you can figure it out, the structure. There's a book by Murphy if you don't know all of those stuff. So the real sample that Ángel was talking about, serum sample, microalgae. If we confronted this to a NeoMS library, we can annotate 46% of the total amount of the of what? So 50, I don't remember now what was 46% of stuff represent. This is something, this 56% of
the compounds that are in our library.
Yeah, the compounds that are in our library, they are being annotated in the serum samples, right?
Exactly. This is the
right.
Among the annotated compounds.
Right.
We annotate the 40% of the compounds in our library and the 44% the compounds of the GMPS library.
Yes.
But
so basically, you can use real samples or real data, or real biological information to retrieve non-compounds. That's the replication process. And like Ángel was saying, you can also try to retrieve unknown information. For example, what we found in some of the microalgae, I think it was, is like we found a metabolite of pinolenic acid. Yeah, that's famous stuff in the pine oil. Well, I don't know if there is any pine oil ever found in microalgae. Maybe it's something to look for. We have as well found some adrenic acid stuff in the serum, which is maybe logical. But the funny thing is that none of the pinolenic acid nor the adrenic acid are in OXPUFA at all. But you can retrieve, therefore, oxylipins or neopUFA that were not there at the beginning in your OXPUFA samples. that you work at the beginning. So you don't have to oxidize probably all the PUFA in the world. So one more little thing. Remember I told you about these Valerie O'Donnell compounds that they had no MS2 data. Obviously, in that paper, in the end, they produced this data. It's not digital, so we extracted this. We created an EMGF file. We did this yesterday night, yeah, just for the talk. And we submitted with our OxPUFA stuff, all the data, and we have a match. We have just like almost a perfect match. So the non-enzymatic oxidation, which was known to produce this epoxy compound, is basically ascertained by that molecular networking thing. And I remember talking with Alan Brash. He wasn't quite so sure about this, but actually I think we have a proof that non-enzymatic conversion can produce that's the epoxy hydroperoxide. So we don't quite know if the double bond is there. Probably the fragmentation will be the same, but definitely the compound is there to analyze. So I think we can conclude this. Sorry if we were a bit fast. We wanted to say so much. There's many stuff that we can improve, or you can improve if you want to use that method. Obviously, you can work on another LC method if you want more separation. If you have fancy machine, you can use ion mobility, right? But what would be nice to do, if there is any organic chemist that is listening to this talk, the one that send emails asking for standard is like, you may be able to implement that neo MSMS library. For example, the epoxy isoprostanoids. They are not available at all in MS2 data. What you can do, let's say that you're a big fan of the dioxolane compounds. Actually, those dioxolane compounds that I talk about, they are really the precursor of a lot of end product of oxylipin, but nobody really used them for quantification, whatever could be a biomarker of anything. Now, if you can find the cluster of this compound in OxPUFA, you can build your MRM method if you want a better quantification. There is a workflow on Skyline when you can export your high resolution fragmentation thing and export into an MRM method, and then you can pick your Q-trap or whatever. There is an equation to use in that process, and you're more or less fine with a semi-quantitative, much better, I guess, method than high resolution. Remember, and this is something that we discuss a lot with Ángel, this is not quite so an analytical quantification workflow that we just talked about there, right? This is more an identification method so far. And I hope you will try it really. I mean, it's not something quite difficult to do, right? You guys are Oxylipins people. You probably have a lot of samples, like a machine, an IDX somewhere in a platform. You should try it. It's something fun to do, a lot of stuff to understand in the fragmentation process and the way. Hopefully, for all this, we have to thank all the people, all the organic chemists that provide these molecules. So all the people, the organic chemists in the lab, the PhD student, the master, the postdoc, and obviously all the team. Anis especially is soon going to be a doctor in a couple of days. We thank all the biochemists in the Jean-Yves Le Guenec, Marie Demion group, Alain Lacampagne too, and all the money, the funding from Spain, from France, and so on, and all the people that we bothered during those 3 years, Porter, Murphy, and all some French chemists as well, and especially dedicated to Jackson Roberts, who died recently. And I hope you all like this talk, and we are ready for the question. Yes?
Yeah.
Thank you.
Thank you very much for this amazing presentation. And we have already one raised hand. Ed, please.
Beautiful talk. As a co-author of the Watrous cell paper you referred to, I'm very impressed with the identification of so many of the unknowns that we had identified in that paper. I wonder if a proper summary of all this is that enzymes are only catalysts. They can only do what chemistry can do, but more efficiently, more specifically, and with some selectivity. So your methods are all non-enzymatic, and in principle, if you have enough chemical reactions, you should be able to mimic chemically sort of everything that biology can do enzymatically. with different efficiencies. And in principle, you could identify all those unknowns that we saw in the Watrous paper and more, and identify everything possible to be made. Are you there now? Or I couldn't follow every detail in the presentation. There's so many molecules, so many fragmentations.
Yeah,
between that and mass spec fragmentation,
yes.
Can you? What percent of the universe can you see?
Right. Okay. Yeah. I mean, that's a big question. I mean, if you go into the Camrix paper, we actually they replicate, I think, all the Watrous compounds, the putative ones that were correlated with inflammation. Yeah. So in the paper, in the supporting information, and even in the text, so in the supporting information, you have all the MS2 explanation, all the fragmentation that we think are related to the identification of those compounds that you're talking about. Obviously, we did not do the 500 ones with Ángel and Anis. But to do this, I guess you have to come up with some software, yeah, some learning machine tools or whatever. But we did by hand the Watrous one with the help of Oxpufa, and all those 9 compounds that he described in the 2 paper, 2019 and the 2019 just after the Daimler paper, where there's 4 or 5 as well important compounds, we basically found the structure and we provided in the paper. And all of them are non-enzymatic stuff, yeah? So they are not, doesn't seem to be any enzymatic relationship with them. Some are just isomer of the 5-HEAT. We think it's an isomer because Watrous had the 5-HEAT as the standard. And he could not see that these other MS2 was as well the 5-HEAT. This is probably related to the network anyhow, because that was not at the beginning of GNPS, but use something else that we use, like the feature-based molecular network. It wasn't there at the moment. So he had this loss of retention time thing that we have now. But anyway, the fragmentation are the same. So we think that if it is a 5-HEAT that he didn't find because he had the standard, he should have seen it, is basically the 5-HEAT that comes from the non-enzymatic version, which has the EE double bond, right? Not the ZE double bond anymore.
Yes.
This is what we thought. It's in the paper. And I think that for annotation, we discussed a lot with this in Ángel. Software, they don't work. It's going to be, it's a mess because the MS2 fragmentation has to happen at some point, but then it has time to happen, yeah? It's milliseconds before it fragments sometimes. The quantum mechanics calculation and so on, they do this in picoseconds, so they cannot mimic the isomerization, the Disalder reaction, whatever it's a Disalder or not, there's debate, right? But anyhow, to explain most of the stuff, Serhan showed it nicely as well in the resolving, you have to have sigmatropic shift of the double bonds. This is impossible to simulate. So what you have to do machine learning, right? So we have Oxpufa, untrained. Oxpufa is not trained. It's all the PUFA in the world, all the MS2 in the world associated to one particular precursor with an MS2 and an MS1, sorry. And you know the molecular formulae, you don't know the structure. But then you have the trained one, which is NeoMS and all the standard you can buy in the world. So you have a trained and an untrained spectra thing. This is how machine learning is working. You will train your machine to learn how MS2 of oxylipin behave. And then you have a train set where you have the structure and the structure of the fragments. Therefore, you should be able to retrieve annotations. This is what we believe, I think so. Ángel?
Yeah, more or less, yes. With the proper training sets, we will be able to annotate better because at the end, some of the fragmentation of the oxylipins are quite peculiar of the oxylipin.
Exactly. Sometimes difficult.
Exactly. If you don't train the current methods that are for the annotation, automatic annotation, normally it doesn't train with oxylipins data. So now we provide all of this data set that can be used for training the new tools.
There's a way to train now machine learning for oxylipin, we think.
So there is a question from Craig here, so maybe we go to the next one.
Thank you so much, Maria. Wow, Jean-Marie, it's so much fun. Thank you for this. Really, really enjoyed it. I mean, kind of following up on Ed's question, I mean, what's it going to take to be able to take your method to the next level and just be able to have a method to identify, quote unquote, all oxylipins? Do you need to be able to just get the right training set? I mean, if so, is the stuff that Gary's been putting in Metlin with your compounds, is that going to be enough to have the training set? I mean, what is it going to take to bring this to, the next discovery level for all oxylipins?
You mean for annotation, right?
Yes.
Well, not much. I think we are there. I mean, we just have to train it, but we're not quite sure how to do it. I mean, the software out there, there's software, MS2 Query. This is the one that Anis during his PhD used, but not full extent, yeah.
Yeah, if you have the proper MSMS structure, something that we can improve also is the feature extraction with better chromatographic separation. Maybe we can decrease the number of chimeric MSMS spectra, and then we will have better MSMS spectra, because it could be that some of the features are not well resolved, and it's the chromatographic challenge that we have as of always for analysis of oxylipins. This is why maybe the new tools or the ion mobility could play a key role. Now there are a lot of people, I think, connected online that are working on ion mobility and oxylipin now. So something that all of these methods, use all of these methods for non-target things.
And that's what we want to have, is a selective tool to non-selectively identify all the oxylipins in a sample.
Right. I'm pretty confident when you see MS2 Query, how it worked, how they developed this, and they made it train. When you train your set properly, it's very good. You get very good result. Maybe you won't get 100%, specificity in one particular position. Maybe it's gonna be a mistake, but I'm not so quite sure. It's very powerful. I mean, that oxpoofa is like how much? 3,000 feature. So that you have 3,000 feature of MS2 for training already, and it's only 4 PUFA. I mean,
right.
You can bring your own C18 thing, yeah. There may be a bit less of compound, obviously, but
right.
But then we need all the standard we can get. Like I was saying, let's say epoxy isoprostanoids are the one that you're looking for in that particular, disease. Right. I guess that if we don't have any MS2 spectra of it, you won't train your set properly, right? And we don't have MS2 spectra of epoxy isoprostanoid, but
exactly. And that's kind of what I'm getting at, is this, I mean, so in order to...
I don't know if it's going to be enough for a particular family that you don't have.
Exactly. Okay.
That maybe, I'm guessing that yes, it should be able. I mean, the fragmentation process, I don't know if Robert Murphy is there, is kind of... is very recurrent. It's almost the same rules that you have to apply. So if your chemical training set is how it works, it's going to learn rules.
I mean, aren't a lot of the MS2 spectra in Metlin now? I mean, you've been sending your compounds to Gary, and Cayman's been sending them. I mean, there should be pretty decent coverage.
Yeah, in Metlin. Oh, yeah, already in Metlin. Yeah, if you use Metlin for identification of isoprostanoids, the 50 something that we have shown here for the GNPS version of it, they're already there in Metlin. So you will retrieve it in Metlin, but it won't help so far, as much as I know, for Metlin for identification of unknown. I know that Gary wanted to already implement the neutral loss and so on, but not quite yet. I think that cosine replication thing. Maybe I'm wrong, yeah, but I don't think it is there yet.
Yeah, you can do it, but then you have to use it one by one, but in theory it will be at the end more or less the same. Yeah, but it's not for molecular networking, of course, but it's just for matching the MSMS one by one.
Yeah,
you can use it because the data is there. Yeah.
Right, actually, yeah.
I have a very final last one. What about truncations? Like all of the reactions which you show, right? You use this different oxidation system and everything, yeah? This is all a full-length oxygenation product. So, but do you see any like a Hock cleavage beta scission reaction product, since you use this non-enzymatic reactions here?
It should be, yeah.
Yeah.
but we didn't look at them. Yeah. So we're basically cutting the MS2 at the moment. Yeah. We are the MS1. We are not looking at them, right?
But
I mean, it means the truncated one, the HHNE or whatever aldehyde leftover by
but they are not.
fermentation hook process. Yeah, that's what you meant. The oak process.
Yes, but I think many of them might get lost just during the sample prep, because if you dry in speed wax, so the small ones will probably evaporate, but a bit longer one, you know, with longer ones. And, yep.
Yeah, they are not as well in the inclusion list and so on. So
But-
yeah.
So
but
the universe is even bigger, maybe.
we also pick it. So the data is there because
Right.
we collect from 2000 until 300 Dalton. So at the end, it will be some of that cluster in the network that we don't
Yeah.
know because we don't have a standard or whatever.
Yeah, exactly. We don't have standard. We didn't look at them. What we want to do as well, we want to do single oxygen transformation. Yeah. So because there's a lot of oxylipin that will be formed that are not formed under triplet oxygen condition. This is the future as well that we want
Yeah,
to do.
and also the oxygen tension that we are planning to playing with different oxygen tensions and to be able to increase a little bit the coverage of the network.
Yeah, I think this is absolutely amazing work, and since you are sharing all the data, openly, right? I think this is an amazing resource for the community, and I think we all can go back to your wonderful data and have a look and answer the questions which we are looking for. So thank you very much for this great presentation. So since we are at the end of our time, so we have to stop here. I would like to thank you as a speaker. That was really amazing talk, and for everyone who was present. This concludes this month's webinar, and our next webinar is now open for the registration on our website, and it will be presented by the Ruth Andrew. So we hope to see all of you there again. Thank you very much, and have a nice evening, morning, afternoon.
Right.
Bye.